Hello, my name is Tanner Tobias. I am an undergraduate student attending SUNY Plattsburgh, and today I will be talking about the changes on redox status and organelle dynamics caused by cellulose nanocrystals. Let's begin with a little bit of background to our research. Traditionally, cellulose-based nanomaterials, such as cellulose nanocrystals, which I will refer to as CNCs throughout the presentation, have been used in the pharmaceutical industry as a carrier for certain therapeutics. The applications of cellulose-based nanomaterials have not been fully explored. Other potential avenues to explore consist of being an immunomodulator or a potential vaccine adjuvant. In prior research by our group, we have demonstrated that cationic CNCs induce an immune response in a variety of cells, including human peripheral blood mononuclear cells, which I will refer to as PBMCs throughout the presentation. When looking at figure 1 below, we used IL-1 as an indicator of an immune response in our PBMCs. We exposed these PBMCs to CNCs METAC1B and CNC METAC2B, which are the gray and black bars respectively. On the left side of figure 1, we observe that CNC METAC1B and CNC METAC2B express IL-1 across all dosage ranges. CNC METAC2B, however, causes dose-dependent IL-1 expression. On the right side of figure 1, we treated our PBMCs with lipopolysaccharide first and then exposed them to CNC METAC1B and CNC METAC2B and compared the IL-1 expression of these CNCs in combination with the LPS to the IL-1 expression of just LPS alone. Across all dosage ranges of both CNCs, IL-1 expression was above that of just LPS alone. What's interesting about engineered cellulose nanocrystals is that they can interact with organelles and instigate different organelle responses, which include the production of mitochondrial reactive oxygen species, induction of endoplasmic reticulum stress, and disruption of the lysosomes. The versatile properties of the CNCs allow them to be chemically modified to achieve desired immune responses, which makes them an interesting candidate for vaccine adjuvants. The goal of this research project was to investigate the biological impact the CNCs have on PBMCs, and then correlate this biological impact with their physical-chemical characteristics. The hypothesis of our experiment was that as CNCs increase in size, the impact on the organelle activity of the PBMCs will be inversely proportional. Moving on to the methods section, I would like to talk about how we treated ourselves. Our PBMCs were freshly prepped from leukotrap filters that we acquired from Champlain Valley Physicians Hospital. These PBMCs were then incubated for 24 hours and then exposed to our CNCs, METAC and AEM, across dosage ranges of 10, 25, and 50 micrograms per milliliter. After CNC exposure, we incubated for 20 hours and then harvested the cells. The intact cells that we took, we investigated the mitochondrial reactive oxygen species, the mitochondrial membrane potential, and the lysosomal acidification. To prepare our CNCs for use in our biological assays, we began with wood-based CNCs. We altered these wood-based CNCs so that they would have cationic functional groups. After they were cationic, we then sterilized them with the autoclave for 15 minutes at 121 degrees Celsius, and then we characterized the CNCs by size and surface charge using dynamic light scattering and zeta potential analysis. Below in figure 2, the image on the left shows the different monomers that we used during synthesis, and the table on the right side shows the different ratios of monomers used during synthesis. The results from the zeta potential analysis and dynamic light scattering are in figure 3. The gray bars represent non-sterile CNCs, whereas the black bars represent autoclaved CNCs. When looking at figure 3A on the left, wood-based CNCs without any functional groups added carry a negative charge. AEM1A and AEM2A carry a positive charge after modification, but this positive charge then changes to a negative charge after sterilizing via the autoclave. METAC1A, 1B, and 2B all carry a positive charge after modification, and this positive charge remains after autoclaving. As we look at the results from the dynamic light scattering, most of the CNCs kept a constant size before and after sterilization. The only two CNCs that did not display this result were METAC 1A, which increased its size after autoclaving, and METAC 2B, which decreased in size after autoclaving. Organelle dynamics in the PBMCs were investigated by observing lysosomal acidification, mitochondrial reactive oxygen species, and mitochondrial membrane potential after CNC exposure. All graphs from the biological assays are from data acquired by a flow cytometer. In the graph, the y-axis measures the number of events occurring, and the x-axis measures the intensity of the fluorescence occurring. The bar graphs in each section display the average fold change over the non-treated cells. Lysosomal acidification was probed for using lysosensor, a pH-sensitive fluorescent probe. Samples treated with lysosensor were obtained via the flow cytometer's green channel. Chloroquine was used as a control in this scenario. From the flow cytometer plots, each dose indicates lysosomal acidification because the dosage peaks are to the right of the chloroquine. This indicates a higher level of fluorescence in our treated PBMCs. The bar graph reveals that METAC 1A and METAC 1B cause dose-dependent lysosomal acidification in our treated PBMCs. Mitochondrial reactive oxygen species were analyzed using MitoSox. MitoSox is a red fluorescent probe that fluoresces when exposed to reactive oxygen species in the mitochondria. Analysis was done in the red channel of the flow cytometer. Antimyosin was used in a positive control for this experiment. The peak for antimyosin is to the rightmost of the plot, indicating a high level of red fluorescence. One peak from each sample aligns itself on the right side of the MitoSox positive control. This characteristic of the plot displays that the presence of our reactive oxygen species in the mitochondria after the PBMCs have been treated with our engineered CNCs. When looking at the bar graph, CNC AEM2A displays an inverse dose response. Mitochondrial membrane potential is analyzed using TMRM. TMRM is a red fluorescent probe that fluoresces in the mitochondria of a cell when the mitochondrial membrane potential is detected. Analysis of the treated PBMCs was done in the red channel of the flow cytometer. FCCP was used as a control in this experiment. This compound is an uncoupler and decreases the mitochondrial membrane potential. In this analysis, FCCP was expected to be on the left side of the plot because it should not have shown any fluorescence. When looking at the plots of our samples, all peaks appear to the right of the stained PBMC control. The peaks align with the FCCP, indicating a decrease in mitochondrial membrane potential, even though the FCCP control appeared on the right side of the graph. The bar graph did not reveal any dose-specific correlations between our CNCs and the mitochondrial membrane potential. To conclude, I would like to discuss observations and future direction for our group's research. Analysis of the CNC's physicochemical characteristics demonstrated that CNC AEM1A and AEM2A acquired a negative charge after sterilization. When observing the size of our CNC's METAC 1A increased in size after sterilization, and CNC METAC 2B decreased in size after sterilization. Analysis of the biological assays displayed important correlations. CNC METAC 1A and 1B showed a dose-response tendency to increase lysosomal acidification. Mitochondrial reactive oxygen species analysis demonstrated that CNC AEM2A shows a reverse dose-dependent response to decrease mitochondrial ROS, and CNC METAC 1B shows a dose-dependent response that increases mitochondrial reactive oxygen species. Finally, mitochondrial membrane potential analysis revealed that CNCs decrease mitochondrial membrane potential, but not in a dose-responsive manner. To overcome large data variation among the PBMC's preparation, alternative methods to assess organelle dynamics will be necessary. Thank you for listening.